This is a session organized by the CCTS for the Center for Clinical and Translational Sciences. I encourage all of you to go to the CCTS website if you have not already done so. There's a lot of resources that you would find helpful, ccts.osu.edu. Now, the reason we're here is to address some of these changing landscape in what is expected out of an applicant. And there are several notices. We have the numbers here that you can take down, and I believe this session will also be available on a recorded basis on the website, so this will be there, so all the content will be posted. What we would like to do today is bring up what the scope of these changes are, and then to share between us what we know. Some of us have had more direct experience in that the first study sections involving these proposals, where these changes would have been already implemented, met or are meeting these days. So the first study sections are just over. I got some material from what was handed out at the study section by the Center for Scientific Review to the reviewers. So you would see a little bit of how reviewers are being instructed in handling our applicants. And based on that, plus what is already announced by the NIH with respect to expectations, I think put those two together, we should be better positioned to really comply with what is expected. So what I would do is flip through some slides that I put together, more to outline the scope of this discussion. And then after that, and I'll go a little bit fast so that we can, because as I said, this would already be available to you, all this content. So we'll just cover the scope and then get into a discussion mode. I'm sure many of you are also reviewing grants, so we would like to learn from you, take all this knowledge together so that the entire audience is well informed on how to handle their submissions. So what's the basis of all of this? So there is grantsmanship. We all know there is grantsmanship, but then there is also content that the grantsmanship should rest on. So if we don't really pay attention to what is really expected and just be superficial about it, I think we may not do very well. So let's try to understand why all of this is coming up. A lot of people are asking why this additional work, on already very limited 12-page or 6-page, depending on the grant type. So there are increasing concerns about reproducibility of science. And there's variables that are not being disclosed in a manner that they should be, that other laboratories could really reproduce. So if you categorize them into broad categories, these are rigor, transparency, reproducibility, we just discussed. We'll talk a little bit about biosketch. Not a lot of people are doing very well with biosketches. And this whole idea of, let me write a grant, and the last moment, somehow I'll put together a biosketch, I strongly discourage, taking that. It is the other way around. Even when your grant is not written up and you're still waiting for preliminary data, that is a good time to put together your biosketch, because the scope of the proposal you already know. And this biosketch has these 5 thematic areas, how to handle those thematic areas, what to really write there, and then how to support that with publications, so on and so forth. Attention needs to be paid. Biosketch, clearly, we all know, directly does not factor into the scoring system, but when they are scoring the investigator and the team, clearly the biosketch content matters. Inclusion of children is becoming more and more a serious matter, and to that should be added gender. Addressing gender and addressing children, and if no children, why not? A cursory, couple of lines may not work. You really have to have a very strong reason for why children are not being used, and as long as you convince them, that's all right. But just because it is too expensive or you cannot do it and you don't have the population, whatever, may not fly. So this is something that needs to be prospectively dealt with as you are charting the scope of the proposal. Authentication of key biological and chemical resources. We'll come to each of these categories individually. Now this part, now at this time, does not fall within the scoring element, but still needs to be provided, and this part, the authentication part, is an additional page or more. There is no page limit, actually. So this is outside. If you're talking about R1, this is outside your 12-page limit. And this authentication piece, a good way to perhaps handle that is to write up in everything that originates and is unique to your own laboratory. And then, if you're collaborating with other laboratories, take their languages and create some sort of a database of authentication of everything unique that you do. And this is also done way ahead of that crunch time of grant writing. Simplification of vertebrate animal sections. So you will see that previously we used to have this detailed justification of animal count, so on and so forth. So some of that load has been taken off our shoulder, so we save some time there. Rigor and transparency. What are the goals? The goals are primarily public accountability. We are spending public money, and there has to be public accountability. There has to be reproducibility, and to some extent, obviously, large extent, social responsibility. So if things that we do only work in a very narrow set of parameters, and then broadly speaking really don't work, that is in some ways cheating. And if you don't qualify that as you publish that, or you don't qualify that as we are writing the grants, that is not taken now favorably. People are questioning that. So essentially here, they're calling this scientific premise. So in developing a scientific premise, which is, you will see shortly, in the significance section, they're asking for a separate category called scientific premise. And in developing the scientific premise, when you're citing all that wonderful literature that supports your line of thinking or creates, it's very important to really critically appraise as to what's the strength and what are their limitations. What part of that model or what part of that design could change, and then that data is no more relevant to the point you're making. So some critical approach is needed there. As I said, gender is becoming, I should say sex, because I just learned a few days ago, which is already here in the slide, that sex is now to do with chromosome and gender is now to do with behavior. Now, again, I'm just sharing with you what I know, and I would like to people that know otherwise to weigh in. So this was not something that I knew. So now I'm very cautious about when I say sex and when I say gender, and so should other people be when they're submitting their grants. We already discussed authentication, and what you see in that red box down there, pretty much everything but authentication feeds into the scoring criteria. Scientific premise. So what is the scientific premise? We just said that essentially in the scientific foundation of this entire project you're proposing, that essentially, in the significance section for our proposals and for K proposals in the research plan section, there should be, this should be dealt with directly. And essentially, there should be justifications, appropriate work. And they're saying when you're citing appropriate work, the strengths and limitations of those work, not just in a quick sentence, it supports, and then move on, not in addressing the critical aspects. Appropriately identified strengths and weaknesses of prior work. They're holding you to that. Propose to fill a significant gap. This we already know. Nothing new there. I'll quickly flip through these so we have enough time for discussion. Significance section, they're saying if you could really have a specific paragraph, I would suggest, because they're saying so, just call it scientific premise. Who knows what gets missed. So a paragraph or a segment within significance, calling it scientific premise. And the strengths and weakness of supporting data. So it is up to us as applicant to mention, as we see, the strengths, but also identify, as we see, the weakness of the data that we are providing. Now, then we have scientific, that was premise. Now we move forward from premise to rigor. Now it's self-explanatory, what is rigor, but let us see how we get measured for rigor here. So essentially, in yellow, in the bullet points down there, you would see this rigor part essentially is to be addressed like the previous segment we said that significance is the home to address scientific premise. But rigor is to be dealt with in the approach section of a typical R01 or for K proposals in research plan. So group size, which I believe we already knew and we did in the past, analyzing anticipating results, we knew that too. Reducing bias in discussions, it is becoming very clear that this is becoming a big buzzword, bias, 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 and gender bias is a big deal, age bias is another, and all types of bias you can think about. Some particular strain of animal now can only produce the data, others can't. And it's not about all those are null and void, as I see. Again, these are my interpretations, subject to your thoughts and criticism. So as long as you're using that as a great model, all you have to do is identify that these are the limitations of this model, but I'm still using it because it's a great tool to do the following, without which pretty much no single tool. So that you have to call out in the proposal. It's not like if the reviewers found out fine, if they didn't find out, even better. That cannot be the approach. So if somebody finds out that you did not address it, and there is actually a limitation widely known, and you're not recognizing, they'll take down the score. Ensuring independent and blinded measurement. Again, this was also not something that I used to very carefully address previously. Whoever is collecting the data is not really analyzing the data, and this is not about just writing. This is also about setting up the laboratory in that way. About how data collection goes, and then how, analysis goes, that there is actually some level of check and balance. It's not like the same person doing all of, so where is the blinding factor? And there are not just blinding, there are many other tools, both statistical and experimental design tools, which we will not get into today, that can be used. So if you look into this, there are actually technical tools available to address this. It was just that I never held myself to this as, stringently as now I must. Improving precision and reducing variability. And many other times, we did not take this really so seriously because you only have 12 pages, and you're so anxious to share the data that you have and the design that you have that we commonly feel that maybe doing anything else is sort of wasting space. So that's the reason. It's not like we don't want to say. I think all of this, any good laboratory would appreciate that all of this is required for good science, but it is just that as we put those material on the grant, these were not priority. Now they have to be priority. Improving precision and reducing variability. So if different labs, collected the data, calibration of the equipment, if an equipment is made by you, then what were the criteria for really validating the reagent or the equipment, so on and so forth. So that obviously would flow into authentication too, but the two would go back and forth. Including, excluding research subjects, and then managing missing data. So these are categorically provided now that, if I'm reviewing, I will have guidance to look for each of this in the grant. So as an applicant, therefore, I would also have to keep it in front of me as we are writing the grants. Approach. Yeah, gender is an approach, yeah. So, and then of course you can further describe that in human subjects, that's a separate story, but gender and children, both human subject and vertebrate animals, need to be addressed, but more importantly, it has to be already within the 12-page limit as to why you're doing only in females or males and not the other gender, and how does that confound, if it does, the scientific strength of what you're proposing. Relevant biological variables, I already mentioned, this is sex, age, source, weight, genetic strain. Again and again, they're asking, sex is a must-do. It's not like you just can't forget it. And I've been doing all my papers are on male, and therefore I'm doing male is not a good answer. So you have to go to the literature, figure out, in one of the revisions currently I'm doing, they basically, they gave us an 18th percentile, and we're revising it, and the main dinging points were that it was all on one gender. And I had to, now we are finding ways of, introducing two genders, and obviously that doubles the volume of work pretty much. So how do you handle that within the same budget and so on and so forth? So there are many practical issues there too that we really need to consider as we plan a new proposal. Will relevant biological variables be controlled or factored into the study design appropriately? Again, you see gender is coming. I write here, sex is equal to chromosome, gender is equal to behavior. It is not written as clearly. This is a disclaimer because here it says source NIH CSR. That is not a copy-paste from NIH. That is something that I heard, I learned, and I put it in a box because we have to be very careful on how we use those words and so on and so forth. So this I'll skip and move forward. As you see, there's a lot about in the consideration of sex, and all these details, if you go to the appropriate notice, details spelled out. So this is something that we must pay attention to. Authentication, as I said, it does not really now fall under the scoring, neither does it fall within the first 12 pages of the R01. So this is, as I understand, there is no page limit for the authentication part. And if applications are non-compliant, you just don't care, they will not even review the application. So this is not like an unimportant piece, it will not be scored, but the reviewers will get confidence. And what is not written in the website, and what is my interpretation is, that if the authentication is lousy, it may flow over in the reviewer's mind to the approach. So yes, it doesn't score. Authentication piece doesn't score. But if I'm proposing something completely new, and the authentication approach I provided is not really convincing, they'll say, well, I don't think it's a specific reagent, and therefore the data that they're showing is not specific or whatever. And so therefore there's a chance that the approach section could be viewed more weakly. Now, this is also to be, as I said, saved separately. There are no page limits. You make a separate PDF, and they give you an uploading option. So you do that separately. And depending on the grant, in my lab, what we are doing is essentially everything we do, we are trying to do an authentication piece and keep it and then cut paste wherever we're doing grants. So not trying to do that with the grants, and that may be one approach, and others may have other approaches of doing this. Guidance for reviewers. And now this are material from what is being provided to the reviewers. And essentially they're asking that the process must be in place to identify and regularly validate key resources used in the research being proposed and avoid unreliable research as a result of misidentified or contaminated resources. So many times we have seen a particular cell line is not even that cell line when somebody really went into the depth and characterized that cell line, and those things are coming out and bothering a lot of people. And now you have literature of like 5,000 papers on that particular cell line. So what do you do? Is it a waste? So on and so forth. So now people are trying to be more prospective about it. Going forward, related review issues, what NIH is saying is that, of course, page limit did not change, but the fact that you don't have space, the fact that it's very expensive to do that, the fact that your institution does not have the capability to do that is not a good answer as we address this. Okay, so we must be very mindful, and when reviewers are told that, they will hold applicants to that standard. And great work, but we did not match. So whatever can be done and should be done, we just need to find the right partner. So that responsibility is with the applicant. Very quickly, a summary here. So as you look at applications as a scientific premise, all applications subject to that. Scientific rigor, all applications. As we discussed, premise is to be in significance, and rigor is to be in approach. Here they're asking, is there a strong scientific premise for this project? Are there strategies to ensure a robust and unbiased approach? Robust. Everybody should be able. Only my lab can do it. We are such a unique lab. Will not cut. If you publish it, and if you're giving the data out and others, and some secret sauce you withheld, because of which others cannot reproduce, gets held against you or me or whoever does that. So we have to be conscious of that. So it's up to us to really give as much detail as we could to make sure others can. And of course, large volumes of writing cannot be done in the grant, so sometimes it is worth publishing it and then citing that publication because now it's in the public domain. Again, as you see here, sex in research strategy approach, that answers your question, to where the sex should be, that they're asking. This is from the NIH, by the way. So this is sex is in the approach here. Are adequate plans to address relevant biological variables, such as sex, included for studies in vertebrate animals and human subject? And all of these will affect the overall impact score, So obviously, and then authentication, is here. This is a new attachment, and this is no, but my interpretation is allows the authentication will flow over to the score as approach. This is not what the NIH is saying. This is my interpretation and what the observations are. So it is up to you to make up your mind on that one. Reviewing rigor and transparency of research, so mentored career development applications. So these are mentored career development applications, scientific premise, scientific premise for the K applicants must be in research strategy, scientific rigor, research strategy. Where do I include it in my critique? And by the way, this critique piece is for the reviewer, okay? And this piece obviously is for the applicant. This table is being shared by NIH and the reviewers. So the reviewers know what to do, because this is new to the applicant, this is also new to the reviewer. So are there strategies to ensure a robust, unbiased approach? And all of this, like you saw in the previous slide, feeds into the overall impact score. The authentication, as the NIH says, doesn't. Finally, like I was mentioning as we got started, it's very important to take time and do your CV. A quickly put together CV irritates the reviewer, and people make comments that this was just cut paste. Maybe they didn't even read some collaborator's CV, some big name. They didn't even read the grant. The personal statement is completely off track. More importantly, what I'm seeing more and more happening is, as you know, now there are 5 thematic areas. A lot of people are now marking these thematic areas, supported by 4 publications, right? And they're giving a header. Our laboratory was the first to discover that. And then there's this paragraph, and then there are those something. So you're making a claim. And then, if it's a true claim, you get credit. If it's a false claim, then obviously somebody will call that out, and they'll ding you. So if you really have something innovative, which we all must as we go to write grants, it's very important to say what exactly you're claiming is unique to you. So in a nutshell, as a header. Now this you already know, conference proceedings, so on and so forth. Frankly, I have never done that in my entire life on abstracts and posters. I don't put them on CVs. But this is also good for K awardees to put, and folks that have a lot of space remaining, you can put that. But for more seasoned investigators, I think that may be something you can ignore. Manuscripts under development can be mentioned in the narrative, in the personal statement, that I submitted this and is under revision, blah, 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 but cannot be put in one of those 4 after the small essay that you have. Okay, and they are so far saying it is optional, but I recommend that you either create a science CV or my bibliography. I have created one in my bibliography. It takes less than 15 minutes. It doesn't take much time. And then somebody has to upload. My bibliography talks to ERA Commons, the NCBI. So whatever you uploaded there, can be pulled and so on and so forth. So take some time. And these are all good to do when you're not submitting a grant and we are several months away. Get it done. And basically then provide the link as you submit the grant. Science TV, here's the website. Again, here's a link that shows, you just click on it. It's pretty simple. Nothing to be shown here. Like I said, inclusion of children in clinical research is becoming a significant issue. I have seen proposals dinged big time for not doing this. Big time, not like point something. Something could go from a 3 to a 5, which is pretty much out of the realm of everything. So this is really serious. So good news for us is we have Nationwide with us. So if there is a question that should be asked in children, and the CCTS is the home, combined home to this campus and Nationwide, as we all know. So let us utilize the CCTS resources to see how we can access these types of resources available next door. Also, they made a new change, which you may have noticed, which is now children are under 18, not under 21. So you used to be under 21, and now they're defining children under 18. Finally, the vertebrate session used to be pretty onerous, and now it has been certain requirements, such as the description of veterinary care or justification of the number of animals, not necessary. It is recommended that you use the word the AVMA, that you will be following, at least state that you'll be following those guidelines. And then, of course, we have a wonderful EULAR who will give us guidance on what exactly it means should you be starting this type of research. This is just quickly what the reviewer sees. And this is from this study section, this cycle. So all these pages look similar, except that you have this box now. On authentication, that is the red box. All those are similar because everything else we do, for example, the premise piece will come under significance, right? And then the rigor piece will come under approach. So there's nothing new there as printed. But there is a new box here, which we did not see in the past, which is this authentication. And then the reviewers get to score us in that direction. Finally, these are some just slides taken from some graphics that just make things interesting, saying how we are pretty blind to, or sort of inherently biased. There's this inherent bias in researchers, and how that bias needs to be managed. And there are some techniques that they're recommending, devil's advocacy, pre-commitment, team of ribos, blind data analysis. And again, these are not things that you just sit and scratch your head and make up. I mean, these are all their own subject. When I started looking into this, there are actually people that do research just on this. So there are actually articles that will tell you how to. It's not like, oh, I'll not let the grad student talk to my postdoc. And so it's not like that. It's not as makeshift as that. So I would recommend read a couple of those articles. They're very pointed guidance and tools. And then I'm sure within our large campus, there will be some expert on that. And then to sit down with them and get some advice, stuff like that. So to take it seriously and address it in a substantive manner. These are some of the other links that NIH has, and if you have any thoughts, NIH has given this website, this email address called reproducibility@nih.gov. So there is an email account that they have created. So if you like it, whatever comments you have, you should write back to them. So they can take your comments into account as they evolve in this process. That's pretty much what we had. I think with this, we have good time remaining for our discussion. Phil, maybe you could lead that.
I just wanted to touch on a couple of the specifics from the robust and unbiased aspect of the approach that we're all supposed to do, particularly as it relates to animal studies. And if you're at the stage of the hypothesis testing, they ought to be randomized. Your experiments obviously should be randomized, and reporting the randomizing scheme has to be part of your approach. And you want to collect the data and appropriately block the animals according to that scheme. Blinding is also important, including, for example, a blinded assessment of the investigators that are assessing and measuring and quantifying the experimental data, blinded to the intervention if possible. You ought to have a sample size estimation in there and power calculations about how you arrive at that sample size estimation. Data handling, you should have prospectively identified. So before you do it afterwards, in your application, you talk about the rules for stopping data collection in advance, the criteria for including and excluding data prospectively, how outliers are handled, prospectively identify that, and then prospectively identify a primary endpoint. You want to report missing data, as Chandan alluded to, secondary to attrition or exclusion. And you want to report whether or not you've repeated the experiments under different biologic conditions and to see how reproducible they are. So it sounds like a hassle, but if you do it, if you're going to do it like that, it doesn't take a lot of, I don't think, pages to do it, but if you outline this in your approach, these key things, that kind of the things that I discussed, and you can find online some of the things that are expected, it does actually make your approach, certainly your proposals, appear rigorous. And that, as a reviewer, they'll be looking at these things, okay, and that'll be I think that'll score favorably for you when the approach is being scored. Okay, it's randomized, they talked about the blinding, they talked about how they're handling missing data. So if you have those pieces in there, it's going to help you with your score. So I think that's important. Now, if you're at the hypothesis generating, you're not really you're not really hypothesis testing, there's more leeway with regard to blinding and randomization. Maybe you're just looking for initial effect. But make that clear. If you're not going to do that, say we're more in the hypothesis testing stage, and we will follow these up with hypothesis generating stage. We'll follow this up with more hypothesis testing with future experiments if you're not going to be doing the randomization and the blinding. So I think that although it's a hassle, there is an opportunity to show how rigorous you're going to be and to outline it in a detailed fashion, in an organized fashion. So that could score favorably for you. I think we'll open it up to questions.
Yes.
Thank you very much for that. My first question is, in any preclinical work that you do, especially in small animal and large animal that are non-human primates, how important is having both genders in your? How important is both genders in your experimentation? And the second thing is, even for preclinical studies, small animal and large animal that are not human primates, how important is it to have a statistician on board to validate your power assessments and so on and so forth for your NIH applications?
Genders.
Yeah, the gender piece is absolutely important. And you may say that this is such a difficult set of experiments that first we would like to go with one gender and then we'll go do another. So you have to address it head on. And it's not like you have to do both genders anyway. And people will understand that it is done in a given sequence. And if you can have, that cardiovascular risk is higher in a given scenario in women, and therefore we are now starting with women, and we will then move on and do men. Or there's a dearth of literature on women, and there's abundant literature in men. I'm making up. And that's a void that I want to fill, and I want to collect women-specific. But you have to address it head on, though. It's not a matter of have to do both. It's a matter of, reasoning why you chose one over the other.
If you can't do both or have it as a variable, you need to talk about it. Yeah. And I would say from the statistician perspective, I don't know if it's mandatory for preclinical or animal work, but it wouldn't hurt. I mean, you don't have to necessarily have one on the grant, but you ought to have a good plan. And as cohesive as that is, if you can do it with just talking to somebody or do it on your own, I don't think you have to have a statistician. But if you need statistical input, even preparing it without having a statistician as a co-investigator, it might not be a bad idea, because a lot of statisticians are involved with the clinical studies and are used to a lot of the power analyses, whereas the preclinical work maybe not as much.
Thank you.
And as the
Since we're recording this, we're getting the microphone to people.
Ask others that have direct statistician experience or not, to share in. This is not about us telling you what it is. This is about sharing all our experiences. So please, madam.
Well, that being said, I work in the Center for Biostatistics, and we appreciate being involved in the grants and helping you make your studies more rigorous and more solid. So we do preclinical work. We have statisticians involved with animal studies. So call us and we'll help you set all these things up and make sure it's set up correctly.
And there's a voucher program from the Center for Clinical and Translational Science. You can get some funding to help with funding statistical planning.
Do these rules apply to R and K type awards only? What about U type awards, do you know?
U. I did not see anything that specifically talks about U. So far, everything I saw is R and K. That doesn't mean it does not apply to U. Somebody will have to look it up. Yeah. But I can say that it does apply to the P, because I know it does apply to the P. So, and P was not in this table. So, yeah, you have there, and then we have here. Yeah, thanks.
Hi, I'm Chris Day. I'm with the Grants Management Office. And just to follow up with your comment, if you look at the new guidance and the application instructions, instead of separating by mechanism, they're separating by project type. So it goes by research projects, multi-projects. So if your U is focused on research, they have specific guidelines for the research. You would follow those. However, if you're same like with a P, it follows those those application types instead of the mechanism.
We have one down in front here.
My question is, my question is regarding bias. Because if you do clinical research, it's clearly many people involved, so you can establish this coding and specification. How do you solve it for the laboratory research? Because if you do handle animals, you have only one graduate student who obviously knows what they feed them and what is number of cages, etc. How do you handle this description, actually?
So, I cannot tell you what is correct, but I can tell you, what I did based on advice that I got. And, it is, within a mouse laboratory, important to have groups that actually collect the data and hand over to other groups, that then report to the PI after processing the data. As we see, it's not okay for that same group to collect the data in a triage, whatever they felt was. So the entire raw data should be moved over, and then this attrition decision or whatever, triaging data decision needs to be through a separate group. Now, most small laboratories may not have that luxury, and I think we can talk to the CCTS and see what mechanisms may be put in place, to provide such, facilities in place that is accessible to smaller laboratories that may not have this type of a setup. Yeah.
I have a question on the scientific premise, especially now that you have to critically address strengths and weaknesses of published literature. Does that in any way restrict the amount of literature that you have to discuss in your significant section, given the amount of space you have?
There is, of course, no limitation. It is all about what is more critical than the other, and for you to triage what you think is less critical. Therein also lies a competition, in what you thought was more critical, and what you triaged to be not critical is also a basis for, evaluation. Now, what they are asking, though, practically speaking, is as you exit that paragraph, do exit with what you think are the key strengths and what you think are the limitations of prior literature. This is not like somebody else said so, that from your viewpoint, and because that viewpoint will then carry forward as you write the remainder of the grant. There has to be some continuity in principle or position that you take as an investigator. And so it's very important to critically have the last few sentences. They're saying at least put 2 or 3 sentences. They're categorically saying that, which is essentially a summary of a critical analysis of the key literature which you think constitutes the main premise of the project that you're proposing.
Yeah, I just want to add to that. You still are telling a story and presenting an argument. So you want to be unbiased when you're doing that in your literature review, but you still are trying to sell what you're doing. If you present too much of the negative data against what you're saying, that's not going to fly. But you also don't know who's going to review it. If you have a real expert in the field that you're addressing, and they might say, well, you've left out these critical. So, I mean, you have to be balanced still. But at the end of the day, you're telling a story and you're trying to present an argument why we need further funding or why this needs further research. Sure. So that's how you should be summing it up. I've looked at, critical review of the literature suggests that, this needs to be done.
Good point.
And so along those lines, what can you give us some indication of the particular formats that fared well at study section? Formats, aesthetically pleasing formats.
See, none of these grants that I know of, the score that we got like 2 days ago, Daniel, was that following this format? Yeah. So we got a 2 percentile on a first submission of a grant. Anybody wants to take a look, they can take a look. And so it's not like we were very smart when we put in. We just, at the time, when we submitted, whatever we knew, we did with that. But frankly, the way we are seeing it now is more clear when the advice came to the reviewers how to review. So when we started looking at this process from the reviewer's point of view, I think it became more clear to the applicant as to what should be the content. And I think if you looked at that, they clearly said, which section, how much. There's very concrete guidance on how this should be handled. So if you go through this material line by line, you should not be, really, it should not be difficult to know exactly what to do. But a few months ago, when this was not there, that was really very sort of amorphous, very difficult to figure out what to do. Yes.
Will resubmissions that were reviewed under the old system be expected to come up to the new one?
Yes, and that is exactly. So what happened is we had a new submission, and which went in October or so, November 5 cycle, and unfortunately this got applied to it. Although it actually went ahead of time, some reviewer was all excited about this, and all this language got put into the critique. Okay, and now when you talk to NIH, they're going to say it is up to the reviewer. If they felt this was necessary, they can do. There is nothing too early. So you cannot penalize a reviewer, because at that point it is left to the reviewer to decide what is important for them. So we actually already got beaten up on these, and therefore I said it got an 18th percentile, and now we are quickly trying to fix it. But I asked this directly, and the answer is yes. That anything that was not subjected to this, once you are going back in, anything that goes back in after January 25th of 2016 must be compliant with these notices.
Two questions. Either I missed, it was too late, or I didn't address it. Now they allowed us to resubmit many times, right?
Yes.
As long as we want, and there is no introduction anymore. Would you suggest to address reviewer questions? Or
no, maybe we clarify what you just said. What you said is sort of right. We still can only revise, once, okay? But we can, previously, we couldn't take the same grant and go in with a new submission. Now the difference is you can take the same grant that was not funded, A1, and go back. It is not like a A2, A3, A4, A5, A6, it's not like that. It is after A1, it is given a new grant number, but we are allowed to take that was not funded and go back and have another A1, and then go back and have another A1. So that you can do, but it is not like, and when you do go for an A1, every time you go for an A1, you do get an introduction page. It is just that when you go after an A1 into a new grant number, at that point, there is no reference to any prior submission. But that project can still be submitted. Not that I know of, but there is a lot of wealth of expertise here, so please weigh in if you feel otherwise. Yes.
So this is sort of a general cut to the chase question. So if I don't have, authentication of biological materials, I'm in big trouble. I'm trying to figure out, of all the changes that you've seen, you should already have had pretty good statistical ideas. You should already probably have thought about gender inclusion. If you were going to sort of summarize the changes that really matter, the stuff that if I go back and write a proposal I need to have at the top of my head, what would they be? So key authentication, definitely yes, because it's a rule. But a lot of these other just seems like a shift in guidelines. So I guess I'm saying, based on your experience, if I go write a proposal this weekend for the R16 that's coming, or the R21 that's coming up, what do I need to really be thinking about? If you could condense it down to 3 points.
I'll tell you about my practical part. I'm sort of terrified now to write a proposal with a reagent, with which we have never published. Because the burden for a reagent or a probe or a device that is not reported is very high. Because no matter what you write, and I shouldn't say that, but I will because it's the way I feel, you may not be able to convince a critic that this is possible because it didn't go through peer review. And what they say is authentication does not need to be a whole bunch of data, but you have to give them an approach. But if there is some skepticism in the reviewer's mind, oh, I tried that, it doesn't work that way, that will have a bearing on And if that one person in the panel speaks up and says, you know what, I tried that, it doesn't work like that. I'm not convinced. Everybody else that doesn't work on that area will take that person as the expert and be biased in favor of that person and against the application. So I think if we have any such unique, better to publish some paper with it and at least to show that it went through the due process of vetting, so that's one thing, and maybe Phil could add some more.
No, I agree. Now, there could be an opportunity, since that is not scored, right? And it could be addressed. It's not supposed to be part of your impact score. And like you said, if a reviewer is looking at it, it's hard for him to take that out of that. So there could be an opportunity for you to get a good score and actually be able to address that to the scientific review officer, I think, no?
Yeah, so in our case, there's one technique, I'll just use that as an example, laser capture microdissection. So we can, say, take one cell and do a whole bunch of measurements in that cell, and there are reviewers that fail to do so. And they have written a comment that, I want to see quality control of all types of that you did before I really believe that you can do that. So it is their privilege, to take that position. But it does sometimes, not always, affect the reviewer's take on the entire approach. That, well, this is, not really feasible. So it may feed into feasibility. So ideally, I think that it should not affect the score. And maybe in most cases it wouldn't. But if there is a central technique that is so important for the proposal, which is really unpublished or something, and it's in the authentication piece, and if that did not convince, ideally the reviewer should not really penalize the science, but if they did, I don't know what recourse we have. Yeah.
But in any event, a reviewer, even before this, if they didn't believe the technique was feasible or reliable, could say that anyway.
Yes, could show that. Yeah, agree.
Just outside of authentication, what are the step changes in the new policies? This is different, really.
If you're doing this kind of stuff anyway, like randomizing and including gender and giving a balanced scientific premise, then not much would be different for you.
The gender, the sex, for sure.
Yeah, you have to have that.
For sure. I am not familiar with how many proposals. I really paid a lot of attention to that. So for sure, that's a significant change. Other changes are, I think, more of the approach. You have to weave it in. Yeah.
I just wanted to add age also in there.
Age.
So it's sex and age both, because if you are using a mice that is like six weeks old in your experiments, so then it's going to be like a youth, comparable to a youth person. And then you are claiming a disease that is a 40-year-old disease paradigm.
Many reviewers are bringing that up.
That has been an issue for us.
With now 40-week-old mice. So, that's
I can see this as actually being a big issue, because there, at least in our field, there's a lot of animal models that were developed with specific either male, female, or a certain week or a certain age, right? And so if you're proposing, I think I hear what you're saying. If you're proposing to use a well-established model of the disease, which was done in male Sprague dollies at a certain age, you're saying I think what you're saying is that you could still use that if you can justify why that was the model that applies to the population you're interested in? Or does it mean that that model is no good anymore, you have to start including all the males, females, all the age, everything else?
I think it's the interpretation of the data, and you have to be carefully spelling it out. What situation is this data established for? That's what my take on this is. So if you're using a animal model that's only kind of represent a human of 40 years old, then when you're putting your interpretation of the data section, you have to kind of mention that there.
Is that not no longer good enough because it's only a model?
Ideally, you would have to have, another model that is suitable for an older group of animals, ideally. But that also, if you flip it, makes room for new R21s to do that. I mean, if that's the expectation, the new models need to be optimized, and one needs to be doing exploratory, new grants. I mean, so what if there is this method that doesn't apply? So
An opportunity from an obstacle.
All right. I think, I hope you found this of some value, and as I said, this was not meant to be, really precise advice on exactly what you should do. This was more like sharing our thoughts and your thoughts in a common forum. So after this, you will basically read what is out there and do what you please to do. And if you feel that something we discussed today was way off target, I would really appreciate knowing, because then I would feed that, and I would make sure that wherever this is uploaded, that your footnotes are there, that after this presentation it was found the following. And I hope to have a tag on, so because this has to be a live document. This is today, we understand this way. Soon, many of you will get your pink sheet back, and you will know so much more. And so this needs to be a live process, because in order to score well, we have to be on the right side of the line on these compliance matters. Thank you so much for
Maybe we can all get together and compare pink sheets later.
Okay.
Thanks, Chandan and Phil.